Hello, this is the slides I presented to the Tropical Agriculture Association meeting in London on the 9th of April. I would like to thank Terry and David for the opportunity to bring this to all of you. I am Mario Caccamo, I'm the CEO of NIAB, and the focus of my presentations is on giving you an update in where we are today with NIAB and what are the plans for. Just briefly about myself, I was born in Argentina and I am a computer scientist by training. I have been the CEO of NIAB since October 2021, but before that I managed the East Moline site, which is part of NIAB since 2016. I have as well been director of the institute, which was called at that point the Genome Analysis Center, which is today called Erlam Institute, which is in Norwich. And I've been working in applied informatics to agriculture since 2009. This picture here is an interview I had with Palab Ghosh from the BBC on the occasion of the royal assent of the new legislation for precision breeding. Is a point I will touch in. Now, it's undoubtedly that we are going through a time of change in agriculture, and very much to summarize it, we are looking at producing food with less inputs. In a way, we want to make sure that we can reduce the use of fertilizers, we can increase the land that is available for green infrastructure and biodiversity, we want to reduce greenhouse emissions, and of course we want to do this while we adapting to climate change, which is already happening. It's important as well to highlight that we don't want to export the impacts of our food production to all the territories. This is very much the guiding principles in where we are now focusing the innovation at NIAB with the purpose of making a difference. A recent study from Sweden has shown that human activity have already pushed the limits of our planetary boundaries. In particular, when it comes to areas of climate change and biodiversity, we have actually already trespassed those. This to put a bit of context into where we need to reduce the impact of agriculture, in particular when it comes to some of the emissions that have a direct impact on climate change. The recent past show us that this is something that is possible. Already in the UK, we have reduced substantially the emissions from agriculture. And what I thought is very important to highlight today is that by bringing innovation and investing in innovation, we will be able to as well produce more with. It's clear as well, of course, that the climate is changing. March 2024 was actually the hottest March on record. Now, this is not news for us, and it's actually adding to the evidence that has been built over the last decade, in where it's clear that the human activity has had a direct impact on this trend in for example, in this case, raising temperatures, which of course will have a direct impact in agriculture, and we have as well a direct impact in the availability of resources to produce food and other elements that are important for the use of land. And now, the other important element is, of course, the impact of our activities, in particular agriculture, to biodiversity loss. And there is a real opportunity we have today by making sure that we can use the innovation we have at hand in where we could hopefully bend the curve of biodiversity loss to make sure that we can retain current biodiversity and hopefully with time increase. But in terms of what are the positive elements we can bring from agriculture, it's important as well that we recognize the impact of innovation, in particular genetic innovation, but as well agronomy, have had in being able today to feed 8 billion people with very much the same land we used in the late 60s. As we look, for example, in the last 50 years, we have doubled human population, and we still could feed this human population in a successful way by only increasing the land use by a little amount. This is actually great reflection on how innovation agriculture could have a very important benefit to society. We need to understand as well that through the last 50 years we have seen other improvements in society that have been only possible because we can provide safe food that is available today more fairly. For example, as another metric, in the 60s, 30% of the human population were considered to be under the we're going to bed, if you like, hungry. They were starving, and today this is reduced to 10%. Again, it's not acceptable, but we have seen a massive improvement from that 30% in the 60s to 10%. Now, in terms of where we and NIAB in the context of what we can do in agriculture innovation, I use this slide to demonstrate that in our 100 years history, and these are two pictures from two different queens which are planting a tree at NIAB, the reason why we exist has remained the same. It's about de-risking innovation, about protecting food production for all. In terms of summarizing this in our vision, we provide independent science-based research and information, and we always highlight the terms independent and science-based to support, develop and promote agriculture and horticulture, to support the industry to fulfill their potential. This is very much our main objective in the activities we deliver, while we respect the natural environment. Now, a single slide that shows how quickly NIAB has grown in the last 20 years is this one here, in where we have the key milestones. Two messages in this slide is that today NIAB is in a position where we are 3 times bigger than we were in 2004, and through this phase of expansion, where we bring in other organizations being part of the NIAB group, we managed to create a critical mass today that captures the space of innovation in agriculture in a unique way. But the other important point as well in this slide is the diversity of our income, in where we see that through this expansion we managed to add resilience to the organization by bringing income for a number of stakeholders. Some of them are government, but as well we work with industry. Almost half of our income comes today from partnerships with industry. Now, today we are an organization that covers most of the English territory. We have our headquarters in Cambridge, and an important site for NIAB is, of course, our base in East Malling. The turnover of the organization is around £30 million, which, as I said before, is more than 3 times larger than it was in 2004. And we are today recognized as a unique organization that works at the cold face of delivering innovation, with more than 400 staff that includes crop scientists, plant breeders, agronomists, and crop specialists. Now, the unique selling point of NIAB is that within a single organization we can understand the contributions that are made by new genetics, crop varieties, by the environment, be the environment positive or negative. Positive, for example, when we are managing pollinators. Negative, an example, is managing pests, and as well the management of resources like water and fertilizers, to ensure that we can provide a profitable crop for growers in the UK and worldwide. Now, our position as translator is unique, and I use this slide to put this into the context of what we call the technology readiness levels. NIAB leads in areas of pre-competitive, pre-commercial work, from a perspective of being constituted as a charity, enabling the sector, and working with, on the right hand side, our industry partners, and on the left hand side with our academic partners. Now, we would like to be seen as, of course, preferred technology partners for industry, and these are here 2 examples in where we can demonstrate that. One is that we are leading a PhD program in the context of what is called the Collaborative Training postdoctoral program that is funded by the BBSSC, that is in partnership with a number of industry. And another example is our recent transaction with Bayer, in where we transfer our strawberry breeding program, and we're developing through that a very strong partnership with Bayer as one of the leader organizations in our sector. Now, I wanted to spend a bit more time telling you about some of the recent developments in East Malling. And as I said before, East Malling was we was brought to the NIAB group in 2016 as part of that strategy on growing the organization and making sure that we will be consolidating our critical mass in crop. Now, Ismoling is a global synonym for fruit research. This is been founded through some of the work that the site did in more than 100 years ago in developing new rootstocks for apples. Today, for example, the rootstock M9 is by far one of the most popular rootstocks that is used across the globe, and this is the basis for some of the changes we have seen in how we grow apples in intensive production setups. Now, the kind of building principle of when Ismoling was created in 1913 was very much described by this sentence here, which was part of the original articles of the organization, in where it says that the practical needs of the industry are best served by science that investigates the fundamental processes underlying current practical challenges. That's very much aligned with the mission statement for NIAB I mentioned before. Now, one example of more recent activity from Ismoling is the strawberry breeding program. Strawberries is very important horticultural crop in the UK, and on top of that is a high value crop. And what we managed to do in partnership with other research organizations and industries to extend the growing season for strawberries that today in the UK goes from early April to late October. And through that, we have as well managed to focus strongly on the quality of the product. This is a picture of Molin Centenary, which is one of the varieties that emerged from this strawberry breeding program that has been very successful and set the parameters of what is quality for British strawberry production. Some of the key features of this variety is that the uniformity of the shape, the small calyx, right, which you can see in this picture here, which is the leaves part of the fruit here, and how well it has been adapted towards the preference of the UK consumers. Just to give you a number, typically we exceed the 70 million plants sold of Molin Centenary varieties every year. Now, this variety now is part of the portfolio that was acquired by Bayer, but again, it's something we are very proud. It's something that came from the breeding program. We're also working with raspberries, in this case in a much more international consortium. This is another example when we're working with industry, and we have partners in Spain, partners in across Europe and in Australia. And the way this program is implemented is that through this partnership, NIAB retains the ownership of the genetics, but when we get royalty returns, we distribute the returns with through shares with all the partners that have been, well, they're part of the consortium. One of the varieties that has been particularly successful that came from this program is called Molin Vela, and it's a variety that is grown in southern Spain very successfully, and in the UK and northern Europe in the summer months. As you can see here, is a panel of raspberry of the Molin Vela variety. And you can see it today in the supermarket, by the way. It's one of the varieties you will start to see coming from Spain in the early summer, and then as we move into late summer, you will start to see this variety of raspberries coming from UK. The other interesting aspect of the change we have seen in Kent, and is part of our Ismoling, the initiative we have in Ismoling, is the research vineyard. This is focusing on the fast expansion we have seen in the viticulture sector in the UK, in where today we see more than 4,000 hectares being grown of mostly varieties that are suitable for sparkling wines. And we at NIAB in Ismoling have the only research vineyard in the UK. Again, supporting an emerging sector from our perspective of understanding how we can deliver innovation. Most recently, we have as well invested in what we call today the Wine Innovation Center, and this is equipment to be able to assess quality of wines for research. The other important demonstration center we have in Ismoling is the Water Efficiency Technology Center, the WET Center, in where we are focusing specifically here how we can reduce the runoff as a product of irrigation in tabletops grown for strawberries, and as well substrate grown for raspberries, to first of all make sure that we can make better use of the resource, water resource. We reduce as well the impact to the environment by reducing how much is leaking to the environment, and as well support the pursuit of higher quality fruits. And finally, in terms of our recent initiatives in Ismoilin, I wanted to mention the Growing Kent and Medway program that brings together NIAB leading in this project with the University of Kent and University of Greenwich. Growing Kent and Medway was the pillar upon which we built some of the recent investment in the site. And if you haven't seen, or we haven't been to Ismoilin for a few years, you will see the massive improvement on our facilities, from plant growth facilities, including glass houses, as well as some of the new buildings. Again, the intention is, as you arrive to Ismoilin, to be able to have a window into the future in how horticulture crops could be growing in the UK. I kind of leave for you an open invitation for you to come to see the site, but in particular this year we'll have the Fruit Focus event on the 10th of July. Please book that day in your diary and come to see us in Ismoilin. This is a picture of a couple of pictures of the recent developments in Ismoilin. On the left hand side you have the new building. This building is as well hosting the Wine Innovation Center and some of the plant growth facilities. On the right hand side here we have the opening of the new glasshouse complex. These are glass houses that are designed specifically for research purposes. Now that's an overview what we are in terms of the recent investment in Ismoilin. I'd like to spend the rest of my presentation to giving you an update on a couple of other initiatives we're taking forward at NIAB across our presence in the area. Now in Cambridge, we are now working together with the university in what we call the Crop Science Center. This is an alliance that brings together university with NIAB, in where NIAB hosts two professors from plant science from the University of Cambridge in our buildings, in what we are looking into developing synergies which are relevant for three key topics. There is a focus on crop nutrition, improving photosynthesis, and how we can manage and control and protect crops from pests and diseases. Professor Giles Allroyd is the director of this initiative. As I said, he's been particularly successful in attracting funding, philanthropic funding worldwide, and supporting both NIAB and the university to deliver our key objectives in improving or bringing crop science to a better world, as put into the title of my slide, Plant Science into Practice. Now, NIAB is focusing on three translational research priorities. One is genetics, which I mentioned before. The point 2 here, which is improving farm systems, how we can bring elements of agronomy. And the third pillar is how we can improve our access and interpretation of data. Now, in the rest of my presentation, I will give you an example of every one of these, or examples of current projects for every one of these pillars. I will start with breeding. Precision breeding is the ability to be able to further and improve genetics based on new technologies that they are known as gene editing. Now, going back to the basics here, as you will know, plant breeding is based on the principle that we try to select the best plants and cross them with other best plants to try to improve and bring together different traits that could be harbored and could be hosted in a single new variety. We have been taking that approach for more than 10,000 years, very much focused on trying to harness genetic diversity. Right? The intention is, by doing this in a controlled, intelligent manner, we can then select the best traits from different backgrounds, different varieties, to be able to generate new genetics that will maximize our access to improve variety. Now, an example that is very obvious to many people, of course, is how what we have seen in terms of the progress we made with wheat. And one of the traits we have focused in our selection was reducing the height of the crops. Just to make a kind of very obvious example here is a very old depiction of ancient Egyptians harvesting wheat, and you can see that here the wheat is almost actually taller than. This is something through time. Even in the 1950s, we seen this very high wheat. We see people here at NIAB, which is a picture from NIAB, working and examining cereal crops. One of the pillars of the Green Revolution was being able to introduce the dwarfing genes that reduce substantially the height of the crop. Many advantages of reducing the height of the crop is that we focus the biomass production to what we need and try to focus much more of the photosynthesis, if you like, the result of photosynthesis into filling the grains and therefore increasing yields. But by as well reducing the height, we reduce the impact of lodging, right? There is something that is less likely to happen if we have shorter. Now that was, as I said before, the pillars of the Green Revolution that was pushed forward by Norman Borlaug in the late 60s, early 70s, for which we managed to advance massively yields for cereal crops, in particular for wheat in the subcontinent, but as well for rice in The other advantage of breeding and breeding for specific traits is the introduction of what we call this disease resistant or sometimes disease tolerant genes, in where we can make massive improvements in terms of how much a variety could resist current pests, and by doing that we can as well reduce the need for fungicides and pesticides. Now, part of what we have in terms of being able to manage this is that since we understand better the genetic basis and genetic mechanisms that govern all these traits, we can now have much more informed decisions when it comes to some of these crossings, right? Rather than crossing, for a large number of potential combination of varieties, we try to do now a much more direct, much more selective, take a much more direct selective strategy to identify the better variety. Now we can take this to a step further and try to look into how we can then link some of these specific traits to, now that we understand the genetics basis, and in particular because we understand the genome, which is here represented in this string of letters A, C, G's, and T's, we can then make those selections, as I said before, much more directed, better. Now, what genome editing gives us, and it's a tool that has been developed only in the last 10 years, is the ability to make precise modifications in that underlying genome. And by being able to do that, we can reduce the need for a large number of crosses. Still, we cannot dispense altogether from the tools we have from traditional breeding, but we can be much more precise. And one of the technologies that has been recognized recently with the Nobel Prize is the CRISPR-Cas9, which is part of this new approach for precision breeding, as I said before, called gene. Now, just to clarify the distinction between genome editing and GMOs, in GMOs, what we do is we introduce foreign DNA as part of the background of the genetic of the new variety that we're trying to breed. In case of gene editing, there's not introduction of DNA, and by doing that, very much is not distinguishable from some of the tools we have in traditional breeding. The only difference is that we can do it faster and more economically and more precisely. Some examples of what we could achieve with this technology is, for example, controlling the flowering and runnering kind of distinction of the fate of the different tissues in strawberries. It's interesting because if you're working in trying to propagate a variety, you might want to increase the number of runners, but if you, of course, are in a production setting where you're looking at growing strawberries, what you want to do is reduce the runners and increase the flowers. Now, the type of technology that is enabled by gene editing should support us to make this a reality. The other example is reducing the gluten, if you like, which for some people, as we know, it could be toxic, or could make people feel ill, in particular if you are celiac. The intention is to be able to reduce and target, if you like, some of the genes that express these particular proteins to make low gluten wheat that would be tolerant to people that otherwise couldn't consume flour, wheat flours. The other example that is, I think, very well known is being able to reduce the browning in both potatoes and apples. The process that generates or triggers, if you like, this is linked to some genes that are called polyphenol oxidases. It's an oxidation process. And the approach we'll take here is by removing some of these genes, by making them less active. We can reduce the browning with the clear benefits of being able to reduce waste, both in potatoes and apple production, as bruising and the effect of the similar kind of impact between different fruits and different tubers. It could be reduced by not having these genes actively involving the tubers of the fruits. Of course, one of the key benefits will be improved shelf. Now, the regulatory framework in the UK for this for gene editing has recently changed. In March 2023, the Parliament enacted what is now called the Precision Breeding Bill, which very much made that distinction I mentioned before between GM and gene editing. And today we are in a position where in England we could grow gene edited crops in the fields for production. Now, at NIAB we have particularly invested in these technologies. We are experts in the use of genetic transformation, but when it comes to gene editing, we have been at the forefront, and the intention is to be able to partner with other people, and in particular the Crop Science Center, to explore the opportunity that this technology brings for scientifically reasons as well for the production of new. If you want to learn more about the opportunities that gene editing brings to England, I would like you to invite to read my article. This is an article I published last year in Nature Biotechnology on the occasion of the royal assent of the new legislation. Now, this is one of the three pillars I want to tell you. Now, the second pillar is focused on agronomy, and one of the flagship projects we have in the new farming systems is the work we can do in regenerative farming. One of the aspects we champion at NIAB is that regenerative agriculture is very inclusive in the way it's presented, by not having a very strict definition, but it's very much focused in a presentation that follows a number of principles. These principles that probably are well known to some of you, but for example, one is making sure that we can keep the soil surface covered as long as we can, and we reduce and limit as much as we can the plowing of soil, the soil disturbance. We as well introduce a much richer diversity in our rotation, in what we bring, for example, legumes for nitrogen fixation, and we bring brassicas and cereals. We want to make sure as well that through all that we maintain living roots, in where we have the benefit of not just capturing nitrogen when it comes to legumes, but we can push some of the carbon that is captured through these deep into the soils, and when it's possible as well to integrate livestock in the system. It's important that I mention many times when it's possible, and because I think it's very important to recognize that in order to be effective in how we deliver some of these principles, we need to understand the context. It's not the same if you are in a poor soil, or if you're in a very rich soil, if you are in an uplands, if you're in a moor. All of them will require a different approach in making progress in how we can deliver approaches that will protect soil. Now, there is a theory of change behind regenerative farming, which tells us that if we can manage to restore the soil health in the way we approach agriculture, we should be able to improve biodiversity through the implementation of some of these regenerative practices. Our food systems will be more resilient into the future. The intention is to increase the sustainability of agriculture and food systems. We want to, as I said before, improve biodiversity and mitigate the impact that the agriculture has on climate change. Of course, there is a context in which we need to understand this. Here is an example in where a farmer has shown that it's possible, for him it was possible to, in this case, reduce fertilizer substantially without having to have a substantial impact on yields. But the recent paper from Rothenstein where they show that overall, when you look into a number of other, or if you like, larger cohort, more often than not, we will see that by implementing some of these regen practice, they might lead to a reduction in yields, which again, coming back to the principles I mentioned at the beginning of my presentation, as we would like to avoid. Now, one of the examples we have at NIAB is that through the inclusive approach we take to regenerative farming, what we want to see is that there is progress towards practices that will improve soil protection. And this is an example in Headley Hall, which is one of our research stations in the north, in where we have, through a kind of a transition, improved soil protection by introducing practices without having a substantial impact on yield. The other important point to recognize as well is that the exploitation of genotypes historically has been focused on high input systems. One work we are doing with the support from the Morley Agriculture Foundation is trying to look into screening current varieties. Some of the varieties may be even slightly old. They perform in the context of regenerative farming, and trying to see if we can select the ones that perform the best. And finally, another important project we have in the context of regenerative farming is the Center for High Carbon Capture Cropping, where we're looking into diversifying one of the elements I mentioned in some of the principles, the crop rotation, other crops that could increase carbon capture. In particular, in this case, is looking into herbal leys and flower and wildflower mix to try to improve. When we are going through cover cropping, trying to improve carbon retention in the soil and protection of the soil. Overall, our approach to regenerative farming is important that we understand the context, as I mentioned before, and by starting from a baseline that is well understood and is solid, we can measure, we can monitor progress, we can report, and we can verify the validity of our interventions. It's something that which we believe is not currently very widely adopted in the sector, but it's something that is consistent with NIAB's approach, and where we said before that our mission statement is focused on delivering science-based strategies, and this is where we want to take regenerative farming, with all the caveats I mentioned. Now, the final example is on the third pillar, focused on the data, improvement of data and how we interpret the data. And in particular, we are investing in artificial intelligence skills in order to be able to deliver better crops. This is an area that is important for NIAB. Historically, we have been of being able to capture data, but we have never probably implemented enough in terms of data interpretation that we are today able to do with the new technologies. And our approach to these technologies go from the cell level all the way down to the field, and how we can see this delivered in production setting, which is something that is fairly unique to NIAB. Now, this is just an example of the different technologies, but in particular, I wanted to emphasize that for every one of these technologies, there's always a way of translating through the use of artificial intelligence that data that is collected in the field or is collected in the lab into information that we can use for decision. Now, one example I wanted to bring to you is the use of drones to be able to capture field trial data. And this is an example we have in this video here, in where a drone was flown over one of our field trial fields, in where one of the trial fields, in where you see here that every one of these small plots is a different wheat variety. There's a lot of diversity in this one here. You see this as the different density in every variety, but that gives us the space to be able to come with the drone, collect data at the kind of plot level, and this can be processed very effectively and very efficiently for downstream analysis. In this case, this project was funded by the BBSRC and is called Designing Future Wheat. And again, as I said before, it was focused on diversity. That's why you will see that some of these plots are really sparse, just because probably it's a variety or it's a selection that didn't perform very well. If you run the same type of trial with new varieties, you will see better performance across the board. But just to exemplify the type of work we can do with drones, and in particular in terms of collection and interpretation of data. As part of promoting the work we do at NIAB, and in particular here in terms of the work we do in data, we have quarterly release of our landmark issue. This is something which is available in the website. It's available to everyone to download. You can download the publication as a PDF, and let me know if you want to have a printed copy of the landmark issue. But one of the recent issues was focused on the work we do in artificial intelligence to collect data from our field. Now, this is very much everything what I wanted to tell you in terms of who we are today at NIAB. This slide just summarizes some of the key numbers for NIAB and what we are today. I hope you found this exciting in terms of the future, in terms of how we can use new technologies and old technologies for the improvement of crop production, in where we would like to see that in the future we can produce better, more nutritious crops while we protect. Thank you very much for your attention, and as I said before, anything or any question you have, please don't hesitate to contact me, and I'm looking forward to future presentations to the group. Thank you very much for the opportunity.